Okay, grade 11, here we are, lesson number 9, percent change and chi-square. Now we've already done one type of chi-square with our topic 4 unit. We did the quadrat sampling lab. We're going to go over another version of chi-square that you can use for a variety of different differential tests. So first thing, percent change. I'm not going to spend a lot of time on this because it's pretty simple, but it's a really underrated tool for in experimental design. So something to think about, if you're using plants or something like this, it's a really good tool to use. You should definitely be really aware of thinking about, is this going to make sense for my particular experiment or not? So a simple way to figure out how something has changed over time is to calculate the percent change. And the percent change is really good because it's better than a difference because it makes your data points much more comparable. So for example, if you had a bunch of goldfish, like the goldfish example, and they grew, and we see how much they were growing, you could compare their initial length to their final length, and instead of just doing a difference, you can do the percent change. So that way you can tell how much the fish actually grew without having to worry about them all being exactly the same constant starting size. So we're working with anything live, so a lot of experimental biology, of course, work with living organisms. It's really challenging to make sure everything is exactly constant at the start, to make sure that you have a very comparable set of data. So to get around that, we look at the percent change. And the percent change data, that is what we compare to each other and with a statistical analysis. It's not the actual difference in data points. So think about our potato core lab we did, or the dialysis tubing labs we did. Why would that be an ideal choice for those labs? Well, the amount of change is directly tied to the variable. So the membrane area is the one for the dialysis tubing lab, and you can't really easily control that. Like the mass potato core, you make your potato cores all the same size with the cork board, which is a good constant for surface area, but the exact same mass really depends on the density of that individual potato. It's really hard to control that, so instead we do percent change. And look here, the formula, you can write this one down where the new value minus the old value divided by the old times 100. If it's positive, it's an increase, negative, it's a decrease. It's also informative of how it has changed. You can also write it this way, but make sure you write this in your notes because it could show up on your assignment on Wednesday, and it could be a really useful tool for you to use for your actual mock IA and your real IA next year. Okay, let's talk about chi-square. So chi-square allows us to figure out how closely our observed results are to our expected results. So we're comparing observed and expected. And when we do this, we're trying to figure out if the difference between observed and expected is due to chance or some other unexplained phenomenon that's affecting our findings. So if it's due to chance, there's no relationship. If it's just something else, we're learning something or something is affecting our findings here. So a really good use of the chi-square test we haven't done before, we will come back to and do this again next year with genetics, is for Punnett squares. So I know you guys did Punnett squares before in grade 9 and grade 10, I can't remember which one, but we're going to kind of remind ourselves how it works here. So in a Punnett square, we have the dominant and the recessive trait of one parent. This is the father plant, and here we have the dominant and recessive trait of the mother plant. Both of them are heterozygous, and then they cross them. So the dad gives a capital B, mom gives a capital B, this baby is two big B's. The mom gives a big B, the dad gives a little b, this baby is baby little b. This one, the dad gives a big B, mom gives a little b, big B, little b, and they get a little b from dad, a little b from mom, two little b's. So in the symbols here, capital B means white, so it means purple color, and capital B is dominant, so even if it's here showing the white allele, we're only going to see purple, and then when we see white is having two little b's. So this is the expected Mendelian ratio of 3 to 1. When I have two heterozygous parents with one big and one little, crossing a single trait, I expect to get 3 to 1. So I have a theoretical expected ratio that I can test for accuracy using chi-square. Okay, so my expected values are my 3 to 1 ratio here. So the best way I have found personally to do chi-square is you can do with the formula here. I find working in a table makes me much less likely to make a small mistake in my math and therefore make an error in my test, which can be really frustrating. So I do a table where I have the two different outcomes, and I have the observed, and then I have the expected, the difference squared, and then the difference squared divided by expected. So I'm just doing this calculation step by step. And I have a sum happening here. So here's my sigma for sum, and I'm summing over here, and the sum of everything at the end is my chi-squared value. So the overall, this whole table can be where chi is the letter chi. Well, it's not an x, be careful. You have to actually insert the correct symbol if you're using this in a lab. It's chi. O are the observed values, what you actually got from the experiment, and that will always be given to you. Okay? E are the expected values, which are from theory. So in this case, we're using the Mendelian ratio of 3 to 1, which we know from his research and his theory that is what we would expect to see in this particular case. And then, of course, sigma is the sum of all the calculations. So you can write down both the formula, of course, and you can write down a table like this too if you want for your notes on how to do these different tests. It'll be very useful for your assignment on Wednesday. So how do we interpret this? So just like a t-test or any other test, the first step is calculating chi-squared. So in t-test, we've had to figure out what t was and then compare it. In chi-squared test, we have to figure out what chi-squared is and then we compare it. So you have to use a critical values table for chi-squared to figure out if you can reject the null hypothesis. Remember, H naught is a null hypothesis. So you have degrees of freedom. You have to have a critical values table, and you have to, and the degrees of freedom for chi-squared rule is take the number of categories. So how many categories does the data fall into, and then subtract 1 from that. And you also, of course, need to know the p-value. What's the p-value we're going to be using in biology? Always 0.05. So let's do a practice one. So here we have a monohybrid cross. We had smooth and wrinkled seeds, and the parent generation we had smooth seeds, cross wrinkled seeds. The F1 generation we had all smooth seeds. So this is going to be your heterozygous. And then the F2 generation we had 5,474 smooth, 1,850 wrinkled. So that's your observed data. And you want to see if it matches the expected ratio. So here's my table. My two categories are smooth and wrinkled. My observed, 5,474 smooth, 1,850 wrinkled, and here's my sum. My expected, I have to take my total here and use my ratio. So 3 to 1 is my ratio here, where 3 out of this amount is my smooth, and 1 out of that amount is here. So I divide this into 4, then 1/4 goes here, 3/4 goes here. It's my 3 to 1 Mendelian ratio is my expected. The difference, so I'm subtracting them, I have minus 19, I have plus 19, and I have 0, of course. This should always be 0. The difference squared, and then difference squared divided by expected. So 0.6 is I have my difference squared here, divided by my expected. Difference squared here divided by my expected, and then I add them up. I have 0.263. That's my chi-squared value, the sum of these two values here. Okay, so here's my critical values table, and you will need to have this in your notes. I will also give it to you, but it's nice to have all your critical values tables I've been giving to you in your own reference. You can just Google them, or you can use ones from the slides I'm going to give you. It's up to you. But it's nice to have them readily available and you're familiar with them. So here's, of course, our formula. Make sure you have a critical values table for chi-square distribution. So our chi-squared value we calculated was 0.263. How many degrees of freedom? Remember, degrees of freedom is number of categories minus 1. So we had 2 categories. We had wrinkled and we have smooth. So our degrees of freedom is not 2. Look at this. Degrees of freedom is 1. Okay, so is it a good fit? If our critical value here in the table is 3.841 and our chi-squared value is 0.26, is that a good fit? That's a fantastic fit. Remember, this value here, the test value, has to be lower than the critical values table. It has to be lower than this. So if it's much, much lower than this, that's great. We can even look, if we go further, it's going to be even more accurate. Okay, so this is really, really good. We can reject the null hypothesis. There is the expected results does follow the ratio. Okay, your turn. So here we have this data. We cross yellow and green seeds in the parent generation. In the F1, we had all yellows. We know yellow is dominant to green. And in F generation, we had these results. So go ahead, make your table, calculate chi-squared. Do the whole thing. So pause the video and do the whole thing. I want to see this worked out in your notes. You're going to upload that you practice this. Okay, so here's my observed values, 4,400 yellow seeds, 1,024 green seeds, and my sum. I use my 3-to-1 ratio to calculate my 4,518 yellow, 1,506 green. There's a big difference here already. That's a hint. Okay, my difference, 118. Square them, divide by the expected, and my chi-square value is 12.32. That's a lot higher than my last one. So our value is 12.32. How many degrees of freedom? That would be 1. I did it again. 1 degree of freedom. Okay, so our data is not a good fit. It's a lot higher than the critical values in our table, which means we have to accept the null hypothesis, that there is not a match between the theory of the 3-to-1 ratio and the observed results. So this could be due to a variety of factors. Maybe something else is affecting the breeding program. Maybe you don't have enough data collected. Something else is happening here. So that's the first chi-square test. The next test we're going to be doing is called the chi-square test for independence. And this is one you've already done before for our quadrant sampling lab. It's very similar. So in this case, you have a different formula for degrees of freedom. Okay, so make sure you make a note of that. Degrees of freedom is m minus 1 times n minus, so number of rows and number of columns, both times 1, the product of those two things. The same formula here for chi-squared. The difference is we're looking here to see not if it fits a model, but if two different variables are associated or not. So last time it was if the two species can be found in the same place or not. If there's a relationship between the location of two species and the quadrat samples we took on the campus lawn, back when we were on campus. So to use a chi-square test, you're going to use the contingency table, and the contingency table is what we have here, where you're making a table to keep track of the frequencies. So what we're looking for is where this is going to be female, and this is no, and this is female, and this is yes, and this is male, and this is no, and this is male, and this is yes. We're looking for these values here. So we can only use this kind of test if at least frequencies are all at least 5 or more, and the samples were taken randomly from the population. In this case, we don't have expected values from a theory like we have with Mendel's ratio. So this is we have to calculate our expected with this equation. E equals sum of the row times sum of the column over sum of the total. So the way I do this, surprise, surprise, I do lots of tables to help me organize everything. So in this practice question we're going to do, we have a holly tree. So here's a holly tree. And someone has observed that when the tree is growing in the shade, it seems to have smaller berries than the holly tree in the sunny position. And the number of seeds inside the holly actual berries is variable. So they think it's reasonable to predict that larger berries are going to have more seeds than smaller berries. So to investigate this, what they did is they took 60 berries from each tree and they counted the number of berries. That would be a great IA project, actually, looking at something like this kind of ratio. And then, so the two variables are if the tree is sunny or shady, and the number of seeds. So write your null and external hypothesis. So your null should be something like the two variables are independent, the number of seeds is not significantly different in the berries on the two trees. And the experimental, the two variables are associated, there are more seeds in the berries on one tree than the other. You don't even have to say which one's going to have more, so there is a relationship there. Okay, so here are my observed values that would always be given to you, right? So I have my tree in the shade. The number of berries that had 2 seeds was 12. The number of berries that had 3 seeds was 40. The number of berries that had 4 seeds was 8. A total of 60 berries sampled for the shade tree. and I had 60 berries sampled from a sun tree. I had none with only 2 berries. I had 17 with 3 berries. I had 43 with 4 berries. This already looks like there's a big difference here. And I have all my totals. That's my observed value. This will be given to you every time. If not in a table form, at least somehow given to you. So first thing is calculating your expected value. So here's my contingency table, and I have to do sum of the row times sum of the column divided by the sum total. So my expected values, the tree and the shade, I expect to get 6 because I have my sum of my total, 60 on my row, times 12 divided by 120. That gives me 6. I have 28.5 here, so I have 60 times 57 divided by 120, by 20.5. 60 times 51 divided by 120. And here's my total. Again, we'll be adding up to 60. Tree in the sun, 12 times 60 divided by 120, 6. 57 times 60 divided by 120, 51 times 60 divided by 120. Oh, look, they're exactly the same. Surprise, surprise. And my totals here. Okay, so now I have my expected values I calculated here. Remember, in the goodness of fit chi-square test, you're comparing it to a theory, to like a known ratio of predicted value, like with Mendel's ratio. So in this case, you have to calculate your expected. In that case, you are given a ratio to use. Okay, so now I've taken both those tables, combined them into one, where I have observed and expected for the shade tree, observed and expected for the sun tree. And I have the number of seeds per berry and my totals. So to calculate my actual chi-squared value now, I have my observed values, 12, 40, and 8 for the tree and the shade, okay? Expected, take my difference, and here's my chi-squared value for tree and the shade. Okay, and then I want you to do the same thing for tree and the sun. So I did here tree and the shade. You do the same thing for tree and the sun, and then add up all 3 data points to calculate the chi-squared value. Okay, so the final chi-squared value here is 45.3. How many degrees of freedom? We have 1 row, 1 column. So you had 2 rows, 3 columns. So our degrees of freedom is 2. And are we going to accept or reject? We reject because it's way bigger than a critical value of 5.991. So we reject the null hypothesis. They're related. Okay, so here's an example of a chi-squared exam question from an old IB exam. So here they have an area of a map covered by 2 different species of plants, here and here. And they have a scale bar here. Scale bars are a skill from topic 1. And the first question was, use the scale bar to win the area of quadrant A1 given the units. So A1 would be this, and it's going to be 3 by 3 meters, so 9 meters squared. And then outline how the chi-square test can be used to test for an association between the distribution of the 2 species. So doing a distribution test is part of the skills for the IB, but you don't necessarily have to, you're not going to have to actually do the full test on IB exam. You potentially will on your exam for this course of this semester, for this year, or for the assignment on Wednesday, but in the IB exam you won't, it just takes too long. But you're expected to know how to use it and how to interpret it. Okay, so collecting the data, make sure right now everyone should have their mock IAs, your subject approved. A few of you aren't there yet, and I'll be contacting you and your parents early this week to check in with you on that. If you're working with people, you have to have a consent form. So please contact me if that's your case. And start collecting your data. Okay, remember the whole thing is due on May 27th. That is not actually that far away. Okay. All right, that's your homework, is work on your IA data and of course upload your notes from today's lesson. Hope that was helpful, guys. Take care.